morning, everybody. Today we continue with the Streptococcus chapter. This is where we were last time, yes? This is the sequelae of rheumatic fever, pharyngitis, and the immune complex that destroys other parts in the body, heart, skin, brain, and joints. Again, this is pharyngitis, the most common, which is caused by Streptococcus pyogenes, group A, gas, pharyngitis. The commonest of all pharyngitis is caused by strep A. If you look here, pussy, lots of pus, and swollen tonsils. We have impetigo, and impetigo looks like the one which we talked about in staphylococcal infections. We have streptococcal impetigo and we have streptococcal impetigo, both. So in order to differentiate between them, we have to isolate the bacteria in the lab. Otherwise, it might be a little bit difficult. The same thing here. What happens? The immune complex. And then this immune complex can go to the kidney and destroy the kidney tissue. That's the bad thing about gas infections, streptococcal infections, especially A. Post-streptococcal glomerulonephritis. This is another disease. Just like the other four, in the brain, the skin, the joints, we have this one in the kidneys also. It destroys the kidney tissue, particularly the basement membrane. If you remember the glomerulus and glomeruli, there's the basement membrane. The basement membrane is attacked by this immune complex and destroyed. We call this glomerulonephritis or post-streptococcal glomerulonephritis. A glomerulonephritis caused by the infection of strep A, which was initiated originally in the pharynx and then went down. Then finished part A, and then we go to part B. So this is the 15th edition, and in part B, I think it's almost the same. The objectives are almost the same because this is a continuation. So, but little bit we have explaining the antigen detection. How do we detect the streptococcal infections? Because some of them you just detect them by serology. You don't have to do culture. It can happen to save time. That's one. Molecular methods, PCR. Of course, we are not going to talk about PCR in details because you have them in the molecular diagnostics. Microscopic methods. How do we see Streptococcus under the microscope when we stain them? How do they look? And then culture morphology. How do they look also on the plate? Although we talked about this. Serologic testing. Here we're talking about the antigen, detecting the strep itself. And here we are detecting what? The antibody of the strep. So when we say serology, usually, we are detecting the antibody produced by our body. Antibodies. And when we say antigens, we are talking about the bacteria itself or parts of the bacteria. Identify proposed flowcharts for diagnosis. Here we continue the flowchart which we started in staph. You remember in the staph, the flowchart, we talked about gram stain, catalase, and so on. Here we will continue all this in this part. Again, this is what I showed you in part A. We are interested in streptococci in general, but we are interested in these three red ones in particular because we focus on these as they are of medical importance. Streptococcus pyogenes, group A, Streptococcus agalactiae, group B, and Strep pneumoniae, and sometimes enterococci, sometimes. But these three here, the red ones, top priority. Specimen collection, transportation, processing. Before culture, we talked about all of this in chapter 5, which is the first chapter in this course, specimen preparation. So you can refer to that chapter, or maybe the video down below. Good. So antigen detection. What do I mean by antigen detection? We are detecting the bacteria itself, or part of the bacteria, sometimes not all the bacteria. Maybe part that are variance factors, like protein M, like ASOT, yes, the toxins. We detect, if you detect the toxin of the bacteria, then the bacteria is here. If you detect a surface protein of strep, then the bacteria is here. So you either detect the whole bacteria or just part of the bacteria to tell you that the bacteria is here. So we have gas, we have kits like these, and these kits, they save time as they are quick, and they just directly, sometimes we used to detect this directly from the throat. You don't have to do, in the early 90s, we used to do for the Streptococcus pyogenes group A 2 tests. Culture, throat swab, open the mouth, tongue depressor, and just touch the back of the throat and spread on a culture media. Plus, we used to have kits like these. You touch and you put in a solution tube and wait for some time. If it turns blue, then group A is there. If it doesn't turn into blue, then group A is not there. These tests, they support each other. So here you can just chromatographic assay directly from throat swab. You don't have to go to culture. If negative, then you have to do the culture. If positive, you can stop and report results as group A Streptococci. Group B and S. pneumoniae has similar kits for use with the clinical samples such as urine and CSF. So there are kits for group A, for group B, and for pneumoniae. That just you take it and apply the test on the clinical sample directly, whether it's urine and whether it's throat swab or even CSF. This is just to save time and even efforts and even money, more economic. A rapid detection of Lancefield grouping A, B, C, D from culture. Also, we used to have this. What do I mean? Many times, you get the culture positive. There is a strip on the plate. But which strip is this from the culture? Either you do, let's say, gram stain and the test which we will talk about, or you do not, or sometimes we use the serology for confirmation. You just take from the culture and make processing and use this kit, and it can tell you this is group A, this is group B, this is group C, this is group D, and so on. Just agglutination, like the test we saw in the lab, coagulase, where you see coagulation, lattice coagulation, and this tells you that this is positive. We used to do this, and until now, to double check our results. So there are two kits, either on the culture to confirm your results, or directly from the clinical sample to make diagnosis. Honestly, you need these kits, because a strep is not like staph. It's a little bit a headache, little bit more to isolate, because it's more fastidious than S. aureus. Also, these days we have molecular methods, PCR. And if you remember, we talked about the 16S, ribosomal RNA gene. And we said that this applies to all bacteria. So we have here a list of genes. Usually we target a gene or part of the gene, like 16S ribosomal RNA. It's 1,400 base pairs. In the commercial kits, they don't use the 1,400, or they use only maybe the first 500, 600, and they think this is enough to make the diagnosis. So here 16S ribosomal RNA gene, which is very commonly used, it's used for group B. Not only group B according to the book, but also if you go to the literature, PubMed, you can see that 16S ribosomal RNA also it's used for B, Also, it's used for pneumoniae, for many bacteria. We have other genes, like the CAM factor. What is CAM factor? We'll talk about this in the lab. C58 peptidase, it's an enzyme we talked about as a variance factor, which stops the chemotactic factors from coming. Also, phosphoenol pyruvate protein, phosphotransferase, that's an enzyme. Also 16S, as I told you, this comes on top of the list, and vancomycin resistant genes. So all these genes, 1, 2, 3, 4, 5, and others not mentioned in the book, they are used as targets. However, the most commonly used is this one, the 16S. There are also kits that want to save time and effort for you, which is this kit. Cefide, this is mentioned in the book. Group B strip, GBS. It makes DNA extraction, which is usually lengthy procedure, plus amplification, DNA amplification, plus identification directly from a swab within less than 1 hour. How about this? All of this in 1 hour to give you good results. Cefide. Of course, definitely there are drawbacks, disadvantages, but you can imagine. DNA extraction, amplification, speciation, identify B or A or D within 1 hour and directly from a clinical sample like a swab. This is something very good. Companies are trying to catch up and make life easier for you and gaining also economic benefits from this. A gram stain. This is a classical. How would a Streptococci appear under the microscope if stained with gram stain? This is how it looks. This is S. pyogenes, and as the word says, strip, which is like a chain of balls. These chain of balls, of course, usually in clinical samples, if you want to make a gram stain from a clinical sample directly, we will not see this long chain here or this long chain. Maybe you will see like this, 2 or 3, 2 or 3. But if you want to see long chains, you have to culture it in a broth like TSB or thioglycolate, and then after this make a gram stain from the broth. Then you can see long chains like these. You can see sometimes the strip as bacilli or cucubacilli. Can you imagine? This is a strip, and sometimes you cannot tell, is this cocci or bacilli? When you have this young cultures, plus if you have antibiotics. If you make culture for a patient and the patient is on an antibiotic course, then you might see this. Cocci, they resemble bacilli. So broth enhances long chain. You want to see long chain? Culture in a broth and make gram stain from broth. You can see them very long. But still, you cannot tell if this is group B or group A. But it gives you nice culture with long chains. Old cultures. Why do old cultures give us bad gram stain? Because cell wall is destroyed, and then crystal violet fails, and then producing gram negative, gram positive, gram variable. Sometimes you see them blue or you see them red or in between. This we call it a gram variable if the culture is very old. It's due to the deterioration of the cell wall. Agalactiae. See, this is strip. Definitely this is taken from a broth, not from a culture, and looks like strips and chains. But as I told you, this is agalactiae, yes, but you cannot tell if this is agalactiae or pyogenes. You cannot tell from the stripping being long chains or the chaining. Here, this is from the gram stain you can tell. If you see here, these are pairs. 2, 2, 2, and 2. We call this diplococci. What do I mean by diplococci? 2. The diplococci is the pneumoniae. Pneumoniae all the time appears like this. Then this is not a group A, this is not a group B, this is pneumoniae. Especially if you isolate this, let's say from place where you suspect pneumoniae, like sputum, for instance, from the lung. So diplococci, or sometimes they call it lancet shaped. like two triangles, all the same description for the same thing. That's pneumoniae. Culture. Which media do we use for culture of Streptococci? Well, we use a little bit similar to the staph. We use blood, sheep blood agar, 5%, chocolate, and then any broth, thioglycolate, brain heart infusion, or even trypticase soy broth, TSP. But for those labs which have good budget and they want to make life easier for them, they go a little bit more advanced. They use better culture media, such as, have a look. Horse blood and sheep blood, they are the same. And even many people, they use human blood in hospitals. Not recommended the human blood, but because it doesn't display the hemolysis like the sheep blood agar or the horse blood agar. So we have, if you remember in the staph, we talked about PEA, which is selective. And it prevents or inhibits the gram-negative, allowing only the gram-positive. But still, this is a problem, because the gram-positive is too many also. So it can give you a little bit of differentiation, but still you will find some problems. CNA, the same thing. We have antibiotics that inhibit gram-negative, staph, and even bacilli. And this may be a little bit better when you inhibit almost everything except your target. This becomes better. CNA, colistin and nalidixic acid. These are two antibiotics. Nalidixic acid is an antibiotic for urine. Still, this is better than using just blood and chocolate. This is better. What we used to do is this. From throat swab, we used to take the throat, open mouth, tongue depressor, touch, and out. Of course, we shouldn't touch the mouth or the tongue. Just touch the back of your throat and back again, or the tonsils. And spread directly, spread on the blood. Then, just for cheap purposes, quicker purposes, sometimes it worked, sometimes not, to be honest. We used to bring SXT, sulfamethoxazole trimethoprim. It's an antibiotic. And put this disc exactly where we streaked. What does this disc do? It inhibits all the bacteria and makes a zone of inhibition. In this zone of inhibition, some bacteria may grow. And this bacteria that grows in this zone of inhibition is Streptococci. That's theoretically speaking. Practically speaking, things might seem difficult than what you talk. That's my experience. And not only that, also streak on the blood agar, disc of bacitracin, which is an antibiotic that is effective against group A, and then 24 hours. If you see zone of inhibition or traces of zone of inhibition, because we have other bacteria, but sometimes you see light zone of inhibition with bacteria inside and outside, but you can tell this is a zone of inhibition for some bacteria, then it's group A. However, as I told you, practice is more difficult than theory. Yes, but any zone is okay. We'll talk about this test in the lab, bacitracin test. Zone of inhibition, any zone of inhibition, it's okay for this test, because this is diagnostic antibiotic, not treatment. This is diagnostic, not treatment. Remember this. But these two tactics we used to use in the lab. But as I told you again, it's not as simple as I explained. Sometimes you find problems. Sometimes you feel that these are not helping you, honestly. Any zone. See? What's your name? Sana. See, any zone of inhibition. Because this is diagnosed. See bacitracin test. We'll talk about bacitracin test in the lab. Now we are going to this one, to this broth. This is a broth, selective media, and for group B in particular, for women who are pregnant, and sometimes we used to have or receive high vaginal swab, HVS. Then this can be used. Todd Hewitt broth. Some people, they call it Lim broth because of somebody scientist. His name is Lim, and developed this in 1982. And this is only for group B. Inhibits normal flora using, look, gentamicin, nalidixic acid, and colistin. So you have 3 antibiotics in this media that kill everything and leave group B. Or some people, they will say, look, nowadays we have chrome agar, and this chrome agar specifically for strep, it can give you color. If there is strep, let's say strep B, then it will give you mauve color. Colors other than mauve, like blue here, then this is not strep B. So chrome agar, you can find it from companies nowadays, commercial companies, for all bacteria almost, including group B. Either you use Todd Hewitt broth or you can use chrome agar. This is for labs that are almost focusing on high vaginal swabs from ladies. That's how it looks. Mauve color, GBS. Group B, Streptococcus. Light purple to mauve. Again, what's your question? It's mauve. Yes, mauve. MAUVE. It's a mauve color, like light purple or like pinky. Yeah, that's a mauve color. What I'm doing here is I'm giving you a flow chart that summarizes everything. You have general, all strep and others, like blood. When you just streak the throat swab looking for group A from inside your tonsils and just streak it on blood, all sorts of bacteria is there. All bacteria is there. So is it going to help you? No, it's going to puzzle you. That's SBA, chocolate, thioglycolate, BHI, and others. This is for basic labs which do not have a good budget for such things. Then we have selective. Selective, look, in selective media, you have options 1, 2, 3. In a lab, you are not going to use the 3 options together. You only use one option, the one you think is better for you. If you are a lab that receives lots of high vaginal swabs for group B, then go for this option, Todd Hewitt or chrome agar. If you are a lab that goes, not only that, you can add serology to your work. Serology, where you can just from clinical sample, you can check if there's strep or not. So nowadays, science has advanced thanks to the companies which are looking for profit, but at the same time they are helping us. So option 1, PAA and CNA, or option 2 for gas, group A, SBA, sheep blood agar with SXT and bacitracin, and then option 3, if you are anticipating or expecting group B, Todd Hewitt or chrome agar. In any of these options, it's very good practice that you have serology in the lab. You say this is group B, it has mauve color, good. Then let's do it serology. If its agglutination is positive, then our work is really good. We proved this by the mauve color and we proved this by serology. It's a good practice that you have serology to confirm your results in the lab. But all are expensive. Everything you add to your lab, it means adding more money, and it means the bill for the patient will be bigger. But the work is going to be magnificent. And look at this, MALDI-TOF MS. This is only in reference labs, very rich labs, government labs, which we talked about in staff areas. MALDI-TOF MS mass spectrometry is for all bacteria almost. So you will see this MALDI-TOF in all chapters with us. I am not going to ask you for a simple lab to buy MALDI-TOF. MALDI-TOF machine is only for government mainly, or research labs, or rich labs. Okay, we have other selective media. As I told you, some labs, they work for staph, for strep A, strep B. Some labs, like in government, They work all in general, medical, and they have good budget for this, and they have all types of media. Of this media in the lab, which we detect enterococci, is the bile esculin agar, BAA. And the test is usually called BET. And we will have this test in the lab. So in order to have an idea about this bile esculin agar, we have to see the BET test. We will talk about this in details in the lab. Maybe 2 labs to come. Where you have, this is the original color of the media, which is light brown or light or orange. But if you see brown or dark brown, then this is going to be enterococci. We'll talk about this in details, don't worry. Or enterococcocell. This is a media or an agar where you see the enterococci black. You see the colonies not brown but black. If in the lab you want to use, and you are interested all the time in your lab for samples where you expect lots of enterococci, mainly faecium and fecalis, both of them the most common, then you can buy, you don't have to buy both. You can buy one of them, the one which you see better for you. Or again, the chrome agar solution. Companies, they made the chrome agar and only for enterococci, because there are some research labs that focus in their lab on enterococci. Others focus on staph. So if you are focusing on one bacteria in your lab, then chrome agar would be a nice solution for you. So here we have chrome agar VRE, which is vancomycin resistant enterococci. And look, it's again pink to mauve if it's enterococci, and others it's going to be blue. That's the idea. Just look at the color and that's all. Incubation conditions and duration. How much are we going to incubate our cultures, and what are the requirements, oxygen requirements? Do we need oxygen, or do we need CO2, or we need both, or we need anaerobic conditions, 0 oxygen? Let's have a look. Again, Streptococci is facultative anaerobe. It means it can grow where there is oxygen, and it can grow if there is no oxygen, both. But usually, when we say facultative anaerobes, which condition do they prefer? They can grow here and here, but without oxygen. They prefer this. They grow faster, and not only that, they display hemolysis better. And if you don't have the anaerobic condition, you can use the capnophilic condition, where there is CO2. Capnophilic 5 to 10, just in a jar, and add a candle, light a candle, put the plates in a jar, close the candle tightly, and just give it 20 seconds. The candle will go off, and then smoke will come out. This will secure good amount, or the desired percentage of CO2, 5 to 10% CO2. 5 to 10% CO2, the Streptococci, it will display hemolysis better. And if there are colony morphologies like crateriform, the depression, you can see it better. Understood? This is preferred by S. pneumoniae. And many times we used to see in the lab the crateriform, the colony, and there's a depression in the middle. I will show you in a minute. 24 hours, maybe you can just leave it even another day to see much better things. If you remember, that's another idea, which is the streptolysin O and streptolysin S. Here we will talk about the streptolysin O. We said streptolysin O is virulence factor, and it works much better when there is no oxygen, anaerobic conditions. And this toxin is immunogenic. What does this mean? It elicits antibodies from our bodies. We talked about this, yes? Here, in order to make the hemolysis much better, what we do is that we just take the inoculating loop and put it on a blood agar and stab. See, stabbing. Stab down. You know what stabbing means? Piercing the surface of the agar with your loop. The surface of the agar usually 4 millimeters, 5 millimeters maximum, and you bring with your loop and stab it and go down until it touches the bottom and up again. Stabbing. You stab the agar, the plate, 1 time, 2 times, 3 times. Why? Because down here below, oxygen is almost zero. So streptolysin O will work better than on the surface. Therefore, it will display better the hemolysis. That's a practice we do if we suspect strep. Stabbing the surface of the agar 2 times or 3 times. Much better to continue also incubation in an aerobic jar. If you don't have it, CO2 jar will do. If you don't have this and that, then aerobic, but you shouldn't expect very nice results. And you are a lab, at least you should have this jar for capnophilic condition. 24 hours. So strep will grow producing, in the absence of oxygen, down here, hemolysin, streptolysin O, which will hemolyze the blood. Anaerobic conditions enhances beta hemolysis. Again, I am not bored of this. I am showing you again the 3 hemolytic patterns. Hemolytic pattern 1 is alpha, partial, color green. Alpha hemolysis, strep pneumoniae as an example, and it's partial hemolysis, and the color is green. Very good. The second one, beta hemolysis. Very nice. Complete hemolysis. Very good. An example, pyogenes, Streptococcus pyogenes. And usually in good conditions, you will see that almost the background of the bacteria almost colorless. No blood at all. And the third one, gamma hemolysis, which means no hemolysis at all, and the plate will appear red. S. pyogenes. Look at the characteristics of colonies. We call it morphology. Shape, it's circular. So it's almost like a coin. Uniform circle. Elevation, convex or raised, little bit, or convex. Glossy or matte. What is a glossy? What is matte? Good. So glossy or matte. Margin, entire. So it's almost uniform, the margin. Small or pinpointed, not like strip. Color, grayish white. So gray, white, in between. Optical, transparent to translucent. So here we have 3 degrees. Transparent, translucent, and opaque. Transparent, you can see through the glass 100%. Translucent, in between, you cannot see. You can see somebody behind the glass, but not exactly. You cannot identify him or her. And opaque, you cannot see. Transparent, translucent, opaque. So here, transparent to translucent, you can see through it. And hemolysis, group A, pyogenes, beta. So if I ask you in the exam, give me an example of beta hemolytic strep, pyogenes, and agalactiae. Both of these are beta hemolytic. That's pyogenes. And that's how it looks, you see. You will get yourself familiarized with the colony morphology if you work in hospitals. Agalactiae, circular, flat. See, flat. This is one of the rarest. Flat, group B, not convex, like a dome. So it's flat and glossy. Margin entire and smooth like this. By the way, this is one good character or one good feature of strep is the pinpointed. Very, very small. in the first 24 hours, you see them very, very small, that every time we used to see these tiny colonies, then very quickly it comes to our mind, this is strep, not like staph. Or sometimes we wait for 24 hours, another 24 hours, and if it's yeast, because yeast starts like this, pinpointed, very small. Then after 48 hours, 36 hours or 48 hours, it becomes big. If you don't want to wait another 24 hours, easy, what would you do? Wet mount. Wet mount under the microscope, saline or distilled water, and if it's bacteria, very tiny circles, and if it's yeast, then very big, greenish fluorescent. Color, grayish to white, the same. Translucent to opaque, this is darker, not like the other one, transparent to translucent. No, this is translucent to opaque, darker. And hemolysis, the agalactiae, the same. Although a little bit, yes, narrow zone, some gamma. The strongest is group A, and then this one, group B. This is how it looks, beta hemolysis. If you want to know if this is convex or flat, nowadays you have your mobiles and you have good cameras. So you just put the plate on the table and look using your mobile with the cam. If you have a good cam, look from above, bird's view, and look from sideways. So you can see if this is, yes, flat or convex, dome-like. You know what dome is? Yes. That's how they look. Not very raised, almost flat. And this is the hemolysis. This is the bacterial colony. These are the colonies, CFUs, with no blood around. They have eaten the blood. This is due to the hemolysis, streptolysin O. Pneumoniae, this one here. This is the third one we are interested in, which causes pneumonia and meningitis. Shape, circular, and elevation. Look, the elevation, it's not raised, it's not convex as most bacteria. It has a cratery form. The cratery form it's like a volcano of the CFU. In the middle of the colony, there is a depression. This depression, we call it cratery form. And this is mainly S. pneumoniae. And we have seen this many times with S. pneumoniae. I will show you an image in a minute. Glistening, shining, margin entire and smooth, small pinpointed. All Streptococci are small and pinpointed, all of them. Grayish, transparent to opaque. So it can be this or that. Hemolysis, this is alpha. This is the main Streptococci that produces alpha hemolysis, partial hemolysis with green discoloration. Another one is viridans Streptococci. That's how it looks. You see, this looks like a crater, depression in the middle. You can say that this is like a circle, yes, but usually this is depression. And this depression is enhanced in a good way if you incubate this anaerobically or capnophilically under CO2 conditions. Look, this is also cratery form. This is cratery form. This is the hemolysis around. Hemolysis, green discoloration. Maybe it doesn't look green here, but in real situation, when you look at it against light, you will see the green discoloration. Enterococci, this is the fourth one, group D, which comes after the first three. Pyogenes, agalactiae, and pneumoniae, these are our interest. If there is a fourth one, then this is the one that we are going to be interested in. Enterococci, or we call them group D. It's circular, like this, convex, high with dome-like, entire and smooth, small and pinpointed as always, creamy to white, transparent to opaque. So it can have this one is usually gamma, Enterococci. But also they say it can be alpha, it can be beta. And it depends, because Enterococci, many species, not only one. Faecium, Fecalis, and many others. bovis. This is how it looks. Yes, raised, convex. It's a little bit higher than the media. Gamma hemolysis, you see? Gamma hemolysis, no hemolysis at all. And this is what is known about the Enterococci, that it's mainly gamma hemolysis. This is Enterococcus faecalis, and we talked about faecium, the ones that we are interested in amongst the Enterococci. If you look here, these are the tables. They are in the book. And from these tables, some of them, they have tests which we made and tests that are commonly used and employed in the lab and tests that are only for research or for reference labs. If you see here, S. pyogenes and agalactiae, colony size, large, medium, it's all pinpointed. Maybe it's just relatively. Lancefield grouping, this is A and this is A and this is B. Anginosus, not very much well known. Pyre test, this is positive, this is negative. Just concentrate on the pyogenes and agalactiae. VP, Vogus broscawa. See, Vogus broscawa, it's a test we use mainly in enterobacterials. But here can be used if you are a reference lab or you are a research lab. Hippurate test. This one is agalactiae is positive, pyogenes is negative. CAMP test. This one we are going to have in the lab. CAMP test. CAMP test agalactiae. What is CAMP test? Wait until we reach the lab. Bacitration, more than 15 or 16 millimeters. It's very diagnostic for S. pyogenes. This one, bacitration for S. pyogenes. So in the lab, we are going to talk about also pyre. In the lab, we have a lab on this. We have a lab on this. We have a lab on this. This one, no. This one, we have a lab on it, but not for the Enterobacteriaceae or Enterobacterians, the new name. It's rather for the Enterobacterials. Flow chart. You remember when we talked about the flow chart for gram-positive cocci? Every time we started with gram stain. And here it's the same story. We should start with gram stain all the time. It will be helpful, very much helpful, if you also do the wet mount to rule out yeast. Yeast, when they grow, they start like Streptococci, pinpointed. And then after 36 hours or 48 hours, then the colonies become bigger and bigger. Therefore, easier to make wet mount, even before gram stain, just to rule out that this is not yeast, that you are dealing only with bacteria. With gram stain, you can tell it's gram-positive bacilli, leave it, and then go to gram-negative, also leave it for something else. If it's gram-positive cocci, as we said in the lab, we have to go for catalase. Catalase negative and catalase positive. If it's catalase positive, then we go for the staph, coagulase, and the other tests. If it's negative, catalase negative, then this is Streptococci. Definitely we are dealing with Streptococci. If it's Strep, then you have to look at the hemolysis display or the hemolysis pattern. On sheep blood agar, of course. With sheep blood agar, you can get good results. With a human, difficult to get clear hemolysis. So we have, as we said, sheep blood agar, hemolysis on sheep blood agar. We have 3 types of patterns, or we have 3 patterns here. Alpha, beta, gamma. If we talk about alpha, then we have a test which we call optician test. It's a desk-like bacitration. We will have a lab on this also. Optician desk. Optician desk, it's sensitive or resistant. If it's sensitive, then this is going to be S. pneumoniae. If it's resistant, then it's going to be, you have to do other tests, like the BET test. BET test means bile escalin test. We have a lab on this. If it's negative, then it's viridans Streptococci, viridans. If it's positive, then you have to do another 2 tests, NaCl tolerance and pyre or pyre test. Pyre test, we have a lab on this. If both of them, you can do one of them only. If you like just one of them. If any of them is positive, this or this, then it's enterococci. If it's negative, pyre negative, let's say, it's non-enterococci. Then this is alpha, green discoloration. If it's not alpha, and what you saw on the plate, sheep blood agar plate, you saw complete hemolysis, then this is beta. Beta, then you have to do either of these tests, one of them, or you can use both if you like. This one is quicker, the pyre, but people are using bacitracin. I prefer pyre. If bacitracin is sensitive, pyre is positive. Also, as I told you, we have pyre lab on. Yeah, we have a lab on this here. So if it's bacitracin sensitive or pyre positive, then this is going to be S. pyogenes. If it's bacitracin resistant or pyre resistant, then you have to proceed with another two tests, CAM test and hippurate test. We have a lab on CAM test, but not hippurate. CAM test, if it's CAM test positive, then it's agalactiae. If it's hippurate positive, it's also agalactiae. So you can use either of these. You may use both of them if you like, but one of them is enough. If CAMP is negative or hippurate is negative, then you have to go to BIT or NaCl, just like this one here. NaCl, but this one you use BIT. Any of them or both positive, then enterococci. If they are negative, then non-enterococci. If that hemolysis, the plate, displays no hemolysis, gamma hemolysis, then you have to do BET, NaCl, or and pyre. You can use only one of them. You can say I want to use BET. You can say I want to use pyre, or I want to use three of them. Depends on how much time you have and how much budget you have. Positive for any of them, enterococci. If you are using pyre and it's positive, enterococci. If you are using BET and it's positive, enterococci. If you are using three of them, the three have to be positive. Then it's enterococci. Otherwise, it's non-enterococci. You can look here. Very important thing at the end is that gamma hemolysis, they end up with enterococci, non-enterococci, this is most common. But beta hemolysis, if they give you negative on all these tests, then they end up as enterococci, non-enterococci, because we said that enterococci could give us beta hemolysis. The same for alpha. Resistant here, negative here, then you end up with enterococci again, because we said again that enterococci could give us alpha hemolysis, but mainly they give gamma. So this is also very important flow chart. So it's highly recommended that you memorize this and learn it by heart, because it's very important in the diagnosis. Now, as a student, you can memorize it and learn it by heart. But when you are at work, and because you keep repeating the work, because of the much of the practice, because you practice it a lot, then you are going to memorize it. But for the sake of the examinations at the end of this course, you have to have this in your mind. These are just the abbreviations you saw: CAT, catalase, SBA, sheep blood agar, hippurate test, BET, bilescurin agar. Let's say enterococci like fecalis, faecium, durans, and avium. These are the ones. Non-enterococci like group D, like bovis and equinus. So just are resistant, S sensitive, and CAMP, Christie Atkins Monk Peterson test. These are scientists who made this test or developed this test, and pyre, L-pyridinal and amidase. So these here we will have them in the lab. Pyre, CAMP, and others. Okay, BIT also, we will talk about them in the lab. Again, multi-TOF is included. I am not going to talk very much about multi-TOF because at the end it's a machine. Blood culture from clinical samples or bacteria and then you use this machine multitoph MS, you get the susceptibility and identification plus subtyping and maybe the gene resistance, maybe like VITIC2. Serodiagnosis. For Streptococci, serology has been used, and one of them has been used extensively. It's called ASOT. ASOT, anti-streptolysin O titer. And if you remember, we said that the ASO, anti-streptolysin O, and streptolysin S. The one of them, S and O, the one that is immunogenic, makes the body produce antibodies, elicit antibodies, is the O. It's immunogenic. Because O is immunogenic, then we made use of it as test. So if there are antibodies against anti-streptolysin O, this means that you are infected with the Streptococcus. Therefore, this was a very common test, ASOT test. ASOT, anti-streptolysin O titer test. It's just serology. Agglutination, very simple. From the serum. Your sample is the serum, not the culture. Just take serum from the patient and then separate it, centrifugation, and just one drop with one drop. And then if there is agglutination, then you have and it's positive, then you have the streptococcal infection. If not, then you don't have streptococcal infection. As simple as this. S pyogenes, SLO. DNAs, this is another variance factor. Also, they developed a kit for this, testing kit. Protein M, also T antigen, Pele. Hyaluronidase, the same here. Streptokinase. This is the most common. This is the kit. This is the most common. Anti-streptolysin O test, ASOT. Anti-DNase, anti-hyaluronidase, and anti-streptokinase. All of these are antibodies produced by our immune system in response to DNase, to hyaluronidase, and streptokinase, and M protein. But the one that has been commonly utilized in laboratory diagnosis is this one, anti-streptolysin O. So this is the end of part B, which means this is the end of the Streptococci chapter.